We are now recording. I think we're live. So greetings everybody from Sedgwick Reserve. My name is Kate McCurdy. I'm the resident manager here at Sedgwick. My job is to hit record, so I'm done. And try and quiet the noises in the background. I have had the pleasure of working with Carolyn Williams and her crew for, well, Carolyn will have to fact check me on this, maybe 5 summers and 6. And it's been really nice, mostly because it's very quiet, usually at Sedgwick anyway. I can already hear the crickets in the background. And Carolyn is really vested in not only her research, but in including collaborators and undergraduates and graduate students and bringing our otherwise quiet field station to life during the summer. So we've gotten to know each other. We've gotten to watch her daughter, Liana, grow up and have become friends. And it's been very nice to have Carolyn and her crew when they're around. It's been an extra pleasure this summer to have Lisa Trudell, her PhD student, be in residence during the sort of pandemic lockdown. Lisa threaded that needle and got approval to stay out here for almost 2 months. She's out here collecting data, and she's not allowed to leave until she publishes at least the first draft of her So I don't have a whole lot to share other than if I could swing my computer around, I could show you all kinds of heavy equipment around the field station. You might be able to see the pavilion behind me. There's been a whole lot more concrete added since we saw you guys last. We now have this really great outdoor amphitheater that hopefully we'll be able to use again soon. But for now, we're just projecting for the 2 of us that are here on the patio. Carolyn and Lisa are over in the Tipton Meeting House where they're going to give their lecture. And John Everts for the Natural History Society is somewhere in town who's going to say a few words before Carolyn and Lisa take it over.
Thanks, Kate. I just want to thank everybody for signing in, and it looks like we have a big audience. So crickets are apparently a popular topic, and most of us probably do take them for granted, sort of the background sound. And so to have a lecture about crickets is pretty special. So I'm glad there's a lot of interest. And I'd like to welcome everyone who's signed in, from whether you're a society member or a docent at the reserve or somebody in the public who got our press release. Hope you'll enjoy the evening's talk. It'll be about an hour, and there'll be an opportunity for question and answer after the talk by Carolyn and Lisa. She's going to be helping with the talk. Lisa will be taking your questions, and as most of you probably know, a reminder, you can send them in at any time using your chat function on your computer. And since there are a lot of people, I'm assuming quite possibly there'll be some duplicate questions, so don't feel bad if Lisa combines a couple along the way if they're about the same theme and subject. I'd like to just do a special shout out to the members of our Natural History Society for their ongoing support. I know we're not offering the field trips that we would normally offer, and your membership renewals are important and valued by all of us, especially on the board of directors. We're continuing to seek out interesting and great speakers on natural history topics, and we'll continue to offer 1 lecture at least per month through the end of the year. It's especially fertile ground to work with UC Sedgwick Reserve and with Kate, who's been delivering the goods, so to speak. She's been coming up with great ideas through researchers and ideas she's had to work with us. So a special thank you to Kate and the Natural Reserve System. Members who do renew from the Society, just a reminder that we do continue to pay honorariums to our guest lecturers, and we're in the middle of a massive, I won't say massive, but a big remodel of our website. So we do have ongoing expenses. It's not like we don't appreciate those renewals because they do help us a lot. I think that there isn't much more to add to this, but if someone had asked me, a year ago, well, we're going to have a talk in the middle of a pandemic about crickets, including climate change, I would have said you're out of your mind, but here we are. So it's an interesting point to kind of reflect on. And I'm going to turn over to Kate so she can introduce Carolyn. And I hope you enjoy the talk tonight. We're always happy to get feedback. You can find an email on our website. And again, as Kate said, this is now being recorded. And as soon as it's ready to go and posted on our website, we'll send an announcement out to our members mailing list, which is big, to let you know it's available so you can watch it again or tell your family and friends, hey, you got to see this. It's good stuff. So thanks very much, everyone, for tuning in. And thank you, Kate, Carolyn Williams, and her assistant, Lisa Trudell, for coming tonight.
Right. Well, I will say no more introduction other than before we all started getting ready for tonight's talk, it was just really nice to hear from Carolyn and from Lisa and Carolyn's sister, who's also at academia, and Jay and Sarah Reddy, who are in residence at Sedgwick, just hear people's stories about how they came to be in the positions that they're in and sort of what drew Carolyn into research and how she sort of wound up at Berkeley. So I'd really, Carolyn, I know you have a long talk that really focuses about crickets and your research, but if you could introduce yourself and just sort of share with the audience how you came to be in the position that you're in, because you're really a unique and amazing person, and it's so nice to see you embrace your students and your subject and just science in general. So with that, I'm going to turn it over to you, Carolyn, and share with us what you will. Thanks again for being here.
All right, well, awesome. Thank you so much for the lovely introduction, Kate and John. It's been, I'm sitting here looking out the window at the reserve and seeing the quail walk by, and it's just one of my favorite places on earth. I feel so peaceful as soon as I come in here, and so grateful to Kate for everything she's done to facilitate our work. So I grew up in New Zealand. I'm originally a Kiwi. Well, I'm still a Kiwi, and just loved animals from the get-go. Passionate about animals. I was always having ant farms at my house and catching snails and racing them on cafeteria trays, and that interest in organisms and how they work has sort of carried me through till today. But I was the first one in my family to go to university, and I never would have gotten, known even how to get involved in research if I hadn't gone to a university and done some lab courses. My first project was drying out mealworms and then starving the mealworms and drying them out. And Lisa will be laughing because she knows that's exactly what I still like to do. It was just really fired up my enthusiasm and excitement to learn that mealworms can't actually survive being dried out if they're starved first, suggesting that it was an energetically expensive process, which wasn't known at the time. So that was that first sort of thrill, doing something that nobody had done before and discovering something new, and that got me hooked. So I changed my original plan of becoming a veterinarian and have been exploring my love of science ever since. And it's such a privilege and a pleasure to share that love with students. And I think being in the field is really the epitome of what I love about science. So Sedgwick has provided the opportunity to really democratize that opportunity and bring people here into the field who've often never had a pair of boots on before, or never been hiking even. And seeing their eyes open to the wonder of seeing your organism in its natural environment is, that's just my favorite thing about the job. So I'm an associate professor now, just quite recently, got tenure in the Department of Integrative Biology at UC Berkeley. And Lisa, I'll hand over to Lisa now. You can.
Yeah, hi. So hi, everyone. I'm Lisa Trudell, and I am a graduate student in Caroline Williams' lab. at UC Berkeley, where I've been working to earn my PhD for the past 5 years, and hopefully going to be finishing up pretty soon. And yeah, I'm very, very fortunate to have been able to come out here for several years now, every summer, to work on crickets. And I'm excited to tell you a little bit, or share some of the data and the things that we've been learning about the crickets out here in the field. But when I'm not here in the field, I'm also doing a lot of exciting research in the lab, and I'm really interested in how organisms sort of manage their sort of energy budgets, and sort of what they do when they eat different diets, and when they have different machinery to process their different foods, and how that affects how an organism is able to function. And so that's me.
And you'll see another little person in the picture here down the front handing me a butterfly net. That's my daughter Liana, and that's about the size that she was when she first started coming to Sedgwick. Now she's up to my shoulder and 11 years old and almost a teenager. So a lot of you will be concerned, as I am, about the biodiversity crisis that the Earth is currently experiencing. We're seeing a lot of scary headlines about biodiversity declines, and this recent report that was released by the WWF suggests that we're seeing average population declines of about 60% relative to 1970 across these broad groups of organisms. So that's scary. And it's a variety of different threats that are combining to drive these declines in biodiversity. And the one that we are interested in my lab is climate change, which really synergizes with these other threats, because when you have species and populations that are sort of pushed to their limit through things like habitat loss and degradation and overexploitation and pollution and invasive species, climate change can really be the thing that sort of pushes them over the edge. And insects are a really great model to study because they're really, really sensitive to changes in temperature. So they're very small-bodied, and they're cold-blooded organisms. So their body temperatures are the same as the environment temperature, unlike us and other mammals and birds, which maintain a high constant body temperature, which kind of buffers us to some degree from changes of temperature in the environment. And insect species are declining almost across the board. The very vast majority of species are decreasing. You, like me, may have noticed over the last sort of 20 years since I've been driving, there's just less insects hitting the windscreen. You know, even anecdotally, you can sort of see there's just less insects around in many ecosystems. And this is important both because many of us love insects, but also because insect population dynamics are really important for ecosystems. So the insects form the base of most terrestrial food webs. All of the higher animals eat insects. Out here at Sedgwick, certainly we see toads and mice and tarantulas and snakes all eating the crickets. The things like pests, forestry pests, like this mountain pine beetle outbreak that is shown in the top picture here, can actually drive carbon cycling in ecosystems. These brown trees here have been killed by these mountain pine beetles. And the mountain pine beetles, as the temperatures are getting warmer, they're able to expand their range further and further north towards the poles. So it's really important to understand how climate change is going to impact insects, both in terms of the important friends that we need, like pollinators and things that that we depend on, but also in terms of predicting where and when agricultural pests and forestry pests are going to be around. So one of the central questions that that we focus on in my lab is how do changes in temperature impact individual insects? And can we then take those individual insects' responses and scale them up to understanding how it might impact population dynamics and ultimately the functioning of the ecosystems that we all care about? So one of the things that my research is really focused on is winter climate, and that's because winter climate is changing really, really rapidly. So this, the color scale shows the degree of temperature change in degrees Celsius that is expected in the next 100 years. So it's, for the Northern Hemisphere, it's January, because winter's in January. The Southern Hemisphere is July. So up here in the Northern Hemisphere and in North America, we're predicted to see up to 10 or even more degrees of temperature change over the next 100 years, which is way, way higher than these numbers like 1.5 or 2 degrees of warming that we're told are critical thresholds. And we're seeing warmer average temperatures. The minimum temperatures at night and in winter are increasing. Snow is melting earlier. There's really idiosyncratic changes in precipitation. So some areas are going to get more rain and snow, some areas are going to get less. It's going to sort of intensify that variation in the precipitation. So lots of really rapid climate changes in winter, and we're trying to understand how those are going to impact insect populations. So I've worked on a variety of species. I started out on butterflies and moths in my PhD. Can you see my cursor, by the way? Okay. Worked on a variety of different swallowtail species and some moths as well, the fall webworm. For my postdoc, I switched over to, some of us call it the dark side because it's this medical model species, Drosophila melanogaster, so what's commonly called fruit flies, although taxonomists would tell you they're not really fruit flies, they're vinegar flies, but really useful model species, but the ecology is not very well known, and they're not as pretty as butterflies. And then now another aspect of my research, which I won't talk about today, but is still ongoing, is on this beetle, the Sierra willow leaf beetle, which lives in the high elevations of the Sierra Nevada mountains. And so we use another natural reserve property out there, a couple of different reserves, the Sierra Nevada Aquatic Research Lab and also the Owens Valley research labs to do that work, where we're hiking up into the mountains and surveying the populations of these beetles, trying to understand how changes in snow cover are impacting their population dynamics at that really, it's the southern edge of their range. So I'm a comparative physiologist, so I study how the selection pressure, so changes in temperature or changes in snowfall, whatever the particular system requires, impacts the organism by thinking about how cold hardy are they, how cold can it get before they die, how much energy are they going to use at a given temperature, and then trying to understand how that maps up to the population level by influencing these important aspects of fitness. So do they die, and how many babies do they have? Those are the two basic things that we care about when we're trying to get up to the level of population dynamics. And that can help us to understand important evolutionary questions like, can the insects adapt fast enough to keep up with the changes that they're seeing in a given region? Which species are going to be winners and losers of climate change? When and where are certain agricultural pests going to be more prevalent, or are we going to see pollinators decrease? So these sorts of questions. But I'm really not going to touch on this research today because it's not based at Sedgwick. And today it's a talk about the crickets of Sedgwick, which really represented a bit of a shift in my research program when I started at Berkeley. And Lisa's the person who came on and led up that research right from the start. So as Kate said, we've been coming to Sedgwick for many years now. Our first year was in 2015. You can see my little Liana there, just a baby then. 2016, we had a small crew. That was the year that Lisa couldn't come. She was back in the lab doing a very demanding lab experiment. 2017, we had a big group of researchers. Our colleague Bao Jun from China came over. There's lots of undergrads. These are undergrad researchers here, 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 here. This is a high school student, Priyanka, who came out. 2018, we have Chris. This is a student who came for 4 years, every year of his undergraduate degree. Here he is, 2017, 2018. Another grad student in my lab, Kevin, who comes out almost every year. 2019, we had another big group. We acquired a family dog, Lucky. Another grad student from my lab, Andre, was with us, and this really great collaboration grew out of our time at Sedgwick with a professor from University of Washington in Seattle, Jim Carnegie, who's a very famous, eminent animal physiologist who's studied, worked out in the Owens Valley on kangaroo rats and other desert-adapted mammals. And he came with us into the field for a week, and it was just fantastic. And then this year, which was by far the strangest year yet, you can see we're in our little cohabiting pods with masks on, and we're all 6 feet apart. But somehow it still managed to be everything that we would want a field season to be. So thank you so much to Kate and Liza and Frank and the Sedgwick Natural History Society and the docents. It's been such a special experience to be here through these 6 years, because they were also the 6 years over which I was an assistant professor in my department. So it's been a really important sort of marker of my movement through academia. So the crickets of Sedgwick, we're finally talking about the crickets. So this is the cricket that we work on. You might have heard the sound that it makes. They're starting up right now out there. We can hear them. These crickets are really interesting. They live in the grass all around. They like the disturbed road edges. They're really interesting because there's 2 different morphs. So there's a long-winged morph, which you can see here, the wings extend out the back of the wing cases. And these ones can fly. This is the female, and this is the male. So you can see the female has this long ovipositor. That's how you can identify the sexes. Both of them have these cerci on the back, but the males do not have an ovipositor. But you can see here the wings, it's a little indistinct, but you get used to it when you look at them a lot. There's the wings poking out the back of the wing cases. Over here, the short wing morph, this is the female on the top and the male on the bottom. And you can see there's no wings poking out there. Do I have another cricket slide? Okay, so the thing that we study these crickets for is to understand one of the major axes of diversity that structures life on Earth. This is the concept of pace of life, and it's from very, very big organisms that grow very slowly and live a long time and have very few offspring that they invest a lot in, and the offspring have high survival rates. And down at the other end here, we have organisms that live very fast. So this mayfly only lives a few days as an adult. Bacteria lifespan can be, 24 hours or even less. But they have many, many offspring, and they invest very little in parental care of those offspring. And so we are studying in the crickets a little microcosm of this range of variation. So within these 2 morphs within the species represent what's called a polymorphism, so 2 different morphs of life history strategies. And we've got this figure here that was made by a postdoc in the lab, Rebecca Clark, that shows, like, under the hood what is different about these crickets. So I showed you the short wing morph. They have short wings that don't come out the back of the wing cases. They have non-functional degraded flight muscles, so the flight muscles don't work. And instead, they have these really big ovaries. So these short-winged crickets are reproducing very early in their adult life, from about the 3rd day or so of adulthood. On the other hand, the long-winged morph, with their big, long wings, they have functional flight muscles, and they don't develop their ovaries until after they perform a dispersal flight. So they trade off this ability to fly for the ability to reproduce very early. So it's called a flight oogenesis trade-off. Oogenesis means the egg production. So there's a direct competition between these energetically expensive flight muscles and the energetically expensive ovaries, that they can't do both at the same time. They have to partition those activities by having these 2 morphs. And the really nice thing is, it's like a little microcosm of that spectrum of pace of life, but it's within a single species that you can study under the same conditions with everything else being held constant. So it's a really classic system for studying life history trade-offs, the sort of internal constraints that lead to them falling out on that axis of pace of life, and also for understanding the processes that lead to loss of flight, not only in insects, but even in other animals like birds. So the questions that we're interested in are, first, what are the physiological constraints that cause them to have these different morphs within the population? Why don't all the crickets fly and also all grow ovaries from early in life? Can we understand where that sort of internal constraint lies? Also, how does environmental variation impact the crickets with these different life history strategies? And what are the ecological factors, so the characteristics of the environment that mean that those two morphs are both retained within the population rather than flight being lost, which is the general trajectory?
Yeah, and so start to get at some of these ecological questions, it's really helpful to sort of be out here in the field and sort of understanding what the crickets are doing in nature. And so we use a number of different sort of approaches and methods while we're out here to sort of get a good handle on what the population of the crickets at Sedgwick are like. And so these involve things like walking transects. And so a transect is just simply a designated path. And we can walk at a set pace at a set time, and then look in a given area repeatedly as to sort of what we find. And that's a lot of what I've been doing these past few weeks, is just walking transects over and over again, and I'll show you what that looks like. We do visual surveys at night, where all we do is look at the ground and see what we find. One year I did a large mark recapture study that I won't tell you much about, but this is kind of fun because you just sort of pick up crickets that you find, and you can like put a little paint, or I use paint to mark them so that we know it was an individual that I had previously found and identified. Then you can re-release them and then see how many you find back later. Out here at Sedgwick, there's a really fun set of pitfall traps, which are just buckets in the ground. And these are really, really useful because you can just sort of leave them open and see what kind of wanders in overnight. I couple these pitfall traps with speakers that sort of play a mating song to sort of attract crickets to those buckets. And then I can, and I'll show you some of what that looks like. But we can sort of be like a male cricket, and that is really, really they fall for that trick very, very frequently. And so that's an easy way to have the crickets come to you instead of you looking for them. And so one of the first things that we've been looking at over these past few years is sort of what the sort of frequency of the different wing morphs is like. And so you can see on this table, this is just sort of representative numbers during visual surveys performed at night. And you'll see that every year we see mostly short-winged individuals. So these long-winged individuals are pretty rare. They probably represent about 10% of the population, at least in our visual surveys. And so in addition to looking at just the sort of frequencies of long wings versus short winged crickets, we can also have been tracking changes in population densities. And so this is a graph that is showing you the numbers of crickets that we've seen in a 30-minute survey in each of the 4 years. And so you can see that most of the colors are indicative of the type of cricket that we see. So a short-winged adult, the blue is a long-winged adult, and then the green is a juvenile. And you'll notice that there's a lot of annual variation in terms of the numbers of crickets that we'll find in a given evening. And I will say that these data were taken from surveys that were conducted about a year apart in the last week of July. One thing that's pretty striking is a little bit of what happened in 2018, and I'll talk about that. But one of the nice things about this long-term data set I'm gonna be doing is using some of the weather station data that's out here at Sedgwick to sort of look at if there's any climate variables that I can associate with annual differences in these population fluctuations. And so climate differences in in the season is one possible cause for variation in population size. Another cause is like variation in predator abundance. And so this year, 2018, we actually learned that there was a surprise extra predator that year. And it was in the form of a little parasitoid fly. And so this map is a map taken of each little dot represents a field cricket population site, and the colors, the red is if there are flies present, the greens are if they're not. And this map was drawn in 2012, and at that point, Sedgwick is little dot number 3. There had never been flies seen. And our collaborator, Dr. Wagner, who's at University of Lincoln, Nebraska, he's been coming out to Sedgwick for many years, is actually the one that showed us where to find some of the crickets out here. And he said that until 2018, he had never seen a fly. But in 2018, the flies were definitely here. There's just small little house flies, and they use the male songs to locate the crickets. And they deposit their larvae on or around them. And because they use song to find crickets, only the males sing, and so the males have a much higher infection rate than the females. And once the males get infected, or once any cricket gets infected, the larvae will develop inside the cricket, and then eventually it'll kill the cricket. And we can see that in our data. There was a really, really large shift in 2018, not only in population size, but in the sex ratio of the crickets that we were seeing. And so when we were out at night, we saw barely any males and mostly females. Some good news is that back in 2019, things were looking pretty normal. And then this year, the population is you'll see some data next, is really much, has completely recovered. And so this seems to have been just a small blip, and I haven't seen that many flies. I did see one fly last year, but I haven't seen any so far this year. And so that brings me to what I've been doing this year. So the past few years, we've been getting a picture of what's going on between years, and this year I'm looking at really in depth what's going on within a single summer. And so the first thing that I'm showing here are sort of the shifts in the population age structure. And so when we first got out here, we were seeing a much more higher percentage of juveniles compared to adults. And so that's just sort of showing us that as the summer is progressing, we're moving on towards the sort of like mating season time. The other thing that we're noticing, and this is just 2 of my different sites, and I'm plotting here transects where I just counted crickets every 4 days. And you'll see that as the summer has been going on, I've been getting more and more crickets coming up. And so the population has so far been growing. I'm keeping this up, and we'll see if it continues to grow throughout the summer, or if I've reached some sort of peak and then it starts to climb towards fall. So that's to come. The other thing that I've been doing out here is nightly I've been using those pitfall traps to to attract crickets at night using a call. And that structure, that's what this data looks like. And so I can just place my little speaker next to our bucket, and then I can play a song all night. And the crickets, I come back the next morning and I find a bunch of crickets in there, and then I count what I got. And so what you can see here is that unlike there's the blue line is our long-winged crickets, and then the orange line is the short-winged crickets. And unlike when we go out at night and look at the ground, we see a lot more of these long-winged crickets. And we think that we're seeing a lot of these long-winged crickets because they're using the song to navigate at night when they're flying. And the other thing that we're able to do is sort of look at this nightly variation, and the black line is plotting the evening air temperature. To look at how variation in evening air temperature is associated with their flying. And we'll hear a little bit about some of the importance of temperature and flight next. But one of the surprise things, or the best things I think about being out here in the field, is that you get to sort of, there's a lot of some unsuspected surprises. One of which is that we learned that much like humans don't like noisy neighbors, apparently neither do coyotes. And oh, there's a cricket song. And yeah, and so one of the nights earlier this summer, I had a coyote that sort of was attracted to my cricket box and took out the song calling. And so now Caroline will tell you more about some of the mystery that we sort of addressed more recently about how crickets are able to fly at night.
Great. Yeah, so this story was really a international multi-university collaboration. So the lead author, Baojun Sun, was here on sabbatical from the Chinese Institute of Science. Chris was an undergrad who's been at Sedgwick 4 times. Rebecca is a postdoc in the lab. Lisa, you already know. Kevin is another PhD student in my lab, and then Jim Carnegie is the visiting researcher from University of Washington who came out. So Sedgwick is really a place where international collaborations can come together, and also a place where my lab can come together, and we have this unique opportunity to form collaborations within the lab. So the reason we started to think about how do crickets fly at night is because flight requires warm bodies. So as I said, insects are ectothermic or cold-blooded animals. And yet they're the only ectothermic animals that have evolved powered flight. If you think of most of the other animals that can fly, it's either mammals or birds. And even in insects that fly, we typically see that their body temperature, this is the y-axis here, that's the thorax, is the chest portion of the body. It has to be pretty warm. This is 30 degrees, 30 to 35 degrees at the point where they fly. And this happens across a number of different kinds of insects. So butterflies, they require temperatures 28 to 38 degrees. These Glanville fritillaries are even warmer. Asian longhorn beetles need to have body temperatures of about 30. Even Drosophila, which I talked about before, has to be fairly warm in order to fly. And yet lots and lots of insects fly at night. So the blue line shows the nighttime migrations in biomass in tons of insects that are flying above the UK. This is detected by lasers. So it's about 1/3 of insects' aerial migrations occur at night. And nights are kind of cool. These are the average temperatures May to July, just the averages of 1901 to 2000 around the United States. And you can see in all of these areas, even somewhere pretty warm like Florida, the air temperatures are quite a bit cooler than what insects typically need to have in their body in order to fly. So there are a number of ways that insects increase their ability to fly when it's cool. They might either get more heat from the environment somehow, they might increase their internal heat production or retain the heat in their body better, or some insects can circumvent these limitations and actually enable themselves to be able to fly at low body temperatures. So here's a few examples of the ways that insects do this. So you'll probably all have seen butterflies basking on cool mornings in order to get warm enough to fly. There's dorsal basking where they'll spread the wings out flat, or lateral basking like this coleus butterfly that has the wings together. They'll orient to the sun to maximize the heat gain. Dragonflies also do this. But this is really for daytime active species. This is not very effective at night when there isn't that source of solar energy. So this video shows a moth performing muscular thermogenesis. So here the moth is shivering the wing muscles in order to produce heat, just like if we jumped up and down and did star jumps, then we would warm up through that heat that's produced from our muscles. And you can see here, the color map shows the temperatures of different parts of the moth as it does this muscular thermogenesis. Even though the external body temperatures and presumably the air temperatures are cool, 12 degrees, by the time you get into the thorax where that wing musculature is, they're up to the 28 degrees or so that they need to fly. No idea if crickets do this. It had never been investigated at all. A lot of insects use various mechanisms to resist heat loss. So things like insulation, so having fur or scales, just like we might put on a woolly jumper. Changes in morphology so that they have a less surface area for their volume. And things like countercurrent heat exchangers. So you can see here the warm blood coming up through the thorax exchanges its heat before it goes down out towards the abdominal wall. So there's a variety of ways that insects can increase their resistance to heat loss. So we have our cricket system. It's a model for the evolution of flight. And yet we really don't know a single thing about the thermal biology of flight in the crickets. And so we were sort of scratching our heads and thinking, I mean, it's kind of cool out there. We never like to forget our jackets when we're going out to do the transects, because although it may be hot at 8 o'clock, the temperature really drops pretty quickly. So we've got this really neat system where both of the morphs of crickets are nocturnal. So they're all having to be active in these nighttime temperatures, but only one of the morphs flies. So we can use this neat system to ask, you know, they're all crickets, but these particular crickets have to be able to fly at night. How do they do it? So that's what this little story is about. So first of all, we need to see whether the habitat temperatures that they experience on summer nights actually are challenging to flight in the crickets. Maybe they've circumvented these limitations somehow and so they don't care. And then we wanted to test these different hypotheses that I went through. Don't worry about the fourth one, it's in the paper if you're interested. It wasn't very important. So I'm going to go through some of the data. So these are temperature loggers that we put out in the fields at Sedgwick. The blue is the air temperature. So you can see as the day goes on, temperature goes up and it peaks, you know, through the mid-afternoon till about 6 p.m. And then there's this drop that we feel out there in the field. And this is the period of time where the crickets are active, sort of 8 o'clock onwards, depending on the time of year, of course. Just before dark, they start to become active. So at the time when the crickets are active, air temperatures are dropping from about 20 degrees down to, you know, as low as 12 degrees or so. However, the temperatures on the surface of the ground where the crickets are actually sitting are quite a bit warmer. So they could very well be getting some of that heat from the substrate and retaining it in their body. Even warmer, these are the temperatures of the crevices. So the crickets, one of the reasons why we have to use all these different methods to find out how many crickets are there is because they live underground in these little cracks and crevices and they spend most of their time under there. If you put the thermocouples down into the crevices, those crevices are hot. Even as the air cools, that heat is retained below the ground. So that leads to the conclusion that the thermal environment that the crickets are inhabiting is really variable. This is the road out the back of the ranch house. The color corresponds to the temperature. You can see the scale on the side there. So although indeed many of the areas that they might be are around 18 degrees, they can find warmer areas in their habitat. So it's possible that they seek those out. So the next thing we wanted to see if the temperatures that they experience limit flight. So we picked two temperatures that were sort of the average temperatures at the start and at the end of the activity period, so 25 and 18 degrees. And we took the crickets, Baojun did this work, took them into the lab, and he would basically throw them up in a big bin, and he would mark down what sorts of flight-related behaviors they had. So this is how many of them did a controlled descent, which means that they still came down, but it was their wings were open and they were controlling the path. And then this is the proportion that were actually maintaining a trajectory that wasn't just sort of falling down to the ground. And you can see, unsurprisingly, short-winged crickets never fly because they don't have functional wings. But the long-winged crickets, they perform these flight-related behaviors much more at 25 degrees than they do at 18. And in fact, no crickets showed any sign of that descending flight at 18 degrees. And at 25 degrees, there was definitely increased propensity to fly, but it was still a bit limited. Many of them were still not flying, even at 25 degrees. So the next thing we did, we had this thermal gradient. These are heat lamps on the end of it. You turn the heat lamps on, and you can see here using the thermal camera, we get a gradient from it. These are aluminum, got to remember my American pronunciation, aluminum lanes, and they get very hot up by the heat sinks, and then they cool down towards the end. So you can put one cricket in each of these lanes and basically ask the cricket, how warm would you like to be tonight? And so the crickets have some time to move around in the lanes, and they choose a position, and we call that their preferred temperature. That's what the crickets would like to be, how warm they'd like to be. And we did this on crickets that we caught here at Sedgwick. We brought this gradient down with us and set it up outside the classroom. And we also did it back in the lab to rule out any effects of like what the crickets had experienced right before we caught them. We fed the crickets beforehand. All the trials happened at night. And what we found from those experiments is that these long-winged crickets, which are blue throughout this talk, they actually prefer warmer temperatures by a few degrees than do those short-winged crickets. And this was consistent whether they were the crickets we caught and tested at Sedgwick or the crickets that had been bred in the lab, although they were the same species and originated from Sedgwick. So it was very consistent. So the long-winged crickets are choosing warmer temperatures. In these thermal gradients, we were also able to measure the temperatures at which crickets spontaneously initiated flight. So you can see here, this is a cricket. These are two crickets here. And these crickets, subsequent to this picture being taken, then took off and flew out into the night. These all happened here at Sedgwick. And we can measure the body temperatures of the crickets by calibrating the camera. And we found that the temperature of spontaneous flight initiation was about 38 degrees, so really, really warm. So even warmer than those butterflies and flies and beetles that I was talking about before. So flight is strongly limited at the sorts of temperatures that we see in the field. They like to be a lot warmer, and they only fly when their body gets up to, mid-30s. So we also caught a bunch of crickets in the field. As we're walking around, we catch crickets, we put a little thermocouple, we pull their little armpit up, we don't hurt them, just put the thermocouple under their armpit, just like you or I takes our temperature. And we found from doing this many, 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 many times that the long-winged crickets are warmer, their body temperatures are warmer in the field. So we find them in warmer habitats and their body temperatures are warmer. So it sort of suggests that they innately prefer to be warmer, and they're seeking out some of those warmer areas in order to elevate their body temperatures. So the really interesting thing that we found is that crickets do indeed perform this muscular thermogenesis, or shivering of the wings. So this is a picture of a long-winged cricket. The wings are blurry because they're actually shaking those wings back and forth. And we see them doing this all the time in the lab. We see them doing it in the field. And the short-winged crickets don't ever shake their wings at all. They never do this wing vibration behavior. And the long-winged crickets do it much more at 25 degrees than 18 degrees. So at 18 degrees, we never see a cricket initiate that muscular thermogenesis. We think they actually can't. They need a lot of muscle function to be able to generate that shivering motion. And the muscular thermogenesis is extremely effective at producing heat. So the crickets at 25 degrees, this is how many of them performed muscular thermogenesis. So 22 out of 37 of them did it. And the ones that did it increased their body temperatures by between 4 and 8 degrees. At 18 degrees, I showed, this is a separate data set, but I already showed you very few of them actually can do it at 18 degrees. Here, only 4 out of 40 crickets. And it's not as effective. They can only produce up to, 2 to 4 degrees of body temperature using muscular thermogenesis. So it would be better if you were a cricket to move somewhere warm, say as warm as 25 degrees, and then initiate muscular thermogenesis, which can get you up that next 10 degrees that you're going to need to be able to fly. And this is just the control crickets that didn't shake, showing that they did not increase their body temperature. We also found that the long-winged crickets cool more slowly. So this is the cooling rate in degrees Celsius per minute. And the long-wing, this temperature here with the dotted line, that was the cooling rate of the incubator, the environment that we held them in. So a cricket that had no resistance to heat loss would cool at minus 0.1 degrees Celsius per minute. And the short wings were actually quite close to that and overlapping. But the long wings, they had some kind of mechanisms to keep the heat in their body, so they cooled more slowly. We don't know what those mechanisms are. That's something that we're interested in following up. So to cap off this little story, how we put it together, how do crickets fly at night? They're seeing air temperatures of about 18 to 20 degrees. They behaviorally thermoregulate. The long-winged crickets prefer warm microhabitats. They're available in the environment. They're in the crevices. They're in the exposed places on the road. We actually see the crickets on the road all the time. If you're here at Sedgwick at night, that's like a prime place to see some crickets. And that gets them up to about, oh, sorry, this means TB, body temperature. This is the ambient temperature. So this, by moving to these warm microsites, they can get up to 23 to 26 degrees, at which point they're in that range when they can start this shivering behavior. So they initiate that muscular thermogenesis, or the wing shivering, which brings them up another sort of 6 to 8 degrees. They resist heat loss while they're doing that endogenous heat production. And now their body temperature's up at about 28 to 36 degrees, and that's the point at which they can take flight. So that is how a cricket flies at night, all work that was done here at Sedgwick Reserve. So just, I had a few closing thoughts that I wanted to finish with. Sedgwick and the other UC reserves play this incredibly important role in allowing long-term research on important ecosystems that cover the whole range of ecosystems that are available in California. And this, the barrier to entry is very low. It's not that expensive to do the research. We can drive in a big 12-seater van. And so it allows us to bring together researchers from all walks of life. We've brought many high school students, we've brought lots of undergrads, my PhD students, even ones that don't work on crickets like to come because it's super fun. And so that, that ability to bring people together and bringing down the barriers enables us to diversify science. And, you know, the bringing together people from all walks of life, from lots of different sorts of institutions, different nations, different backgrounds, that fuels innovation in science. So that's such an important feature that Sedgwick and the other UC reserves plays. And we couldn't do it without public engagement. You know, the science that we do is funded by taxpayer dollars for the large part. It's our responsibility to communicate it back to the science, to the public. And Sedgwick is a place that brings us together with the people in the community who have a stake in and are interested in and can support this research. So thank you all for coming to this talk and for getting involved and supporting Sedgwick. Yeah, and thank you. You know, there's been a long list of cricket collaborators through the years. I'll just point out the person who got me into the crickets in the first place, Tony Zera. He's now retired, but he's worked on these crickets his whole life. Not actually this species, but a Florida gryllus species. Thank you so much to Kate, who's really facilitated all this research, and all of the staff at Sedgwick through the years, and to my lab group through all this time, and to Lisa. And we'd be glad to take questions.
We got a bunch already. Okay. So how do we do questions? I have a list.
Oh, I'm so glad that you have a list, Lisa, because questions have been coming in, and we were hopeful that you're going to step up and try and categorize those and
We have already.
ask them of Carolyn.
Okay. And so I guess I'll start going through the list. I have them sort of grouped as they came in, but if others come up as we're talking, please just feel free to keep them in the chat. And so the first group of questions that sort of arose were just general cricket, I put as like general cricket questions. And so the first was about what determines the different proportions of the morphs. And so to give a little bit, that's a great question, and there's not a single answer to that. Do you want to start?
You can go ahead.
Okay,
Lisa,
so
Lisa's an expert on these crickets, so I'm going to bat a lot of the questions her way.
And so yes, and there's another question that just came in about the genetics of the morphs, and so the wing length trait itself is highly heritable. It has an estimated like heritability of like 0.86, I think, in one of the species, and so you can breed and select for wing morph, but it's also a environmentally mediated trait. And so if you raise crickets in really high densities, that stresses them out a lot. You end up with more of the short wing crickets. If you rear them at higher temperatures, you end up with more of the longer crickets. And so there's a lot of environmental factors that sort of interact with their genetic backgrounds to sort of result in their wing morph determination. And they do interbreed. And so you can breed a long wing and a short wing, no problem. They're the same species. And so that's not a problem.
And there's a little bit of difference there with things like locusts, which also have a polyphenism, that the long wing and short wing locusts. But in the locusts, it will be brought on, the long wing morph will be brought on by crowding, right?
Mm-hmm.
Whereas in the crickets, it's the opposite. You might expect that if they're crowded and they don't have enough food, that they should be long wing to get away. But in the crickets, we actually find it's the opposite, that it has to be really good conditions to produce these long wing crickets.
Yeah, and there was another question about when we raise them in the lab, what are the type of morph ratios that you see? And actually, so when I breed them in the lab, I interbreed. I don't select for morph. So I make up breeding colonies that have half of each of the wing lengths, and I end up with about 50-50 ratios. But one thing that is interesting is that oftentimes, so every year we bring back some crickets directly from the field, and that first generation is almost always entirely long wing. I don't exactly know why, but and then that goes away through time. And there was another question about morphs in terms of generations. And if they're the second generations of the field crickets have more long wings than the first, that's a question that I would love to have the answer to. I don't.
In other species, you see that
Yeah.
the frequency of morph changes throughout the summer with the longwings peaking earlier in the season and then declining and there being more shortwings in the fall.
And I'm trying to keep track of some of that right now this summer, but I'm not quite sure that I was out early enough to get the first generation. But some of the in terms of some of the interesting information from the pitfall trapping that I'm hoping to get, and I'm starting to get a sense of, is sort of the timing of dispersal through the summer. And already I didn't, the data that I showed you was sort of was cut off at like a week ago. And this past week I've started to see declines in the numbers of longwings, and I'm getting a shift in like the ratios of long wings to short wings, and seeing more short wings in my pitfall traps than long wings. And so I think there is some sort of timing in terms of in terms of dispersal that's maybe related to their generation time, because I think that if the crickets fly too late, then they might not have enough time to mate and or reproduce before the winter time. And so I don't know if I'll have the exact answer to that, but that's what we're starting to sort of get some better sense of. There's a question that I would also love to answer about what do crickets eat. I don't know what they eat in the field. They're pretty generalist feeders, and so they eat. I think they typically eat a lot of vegetation. They do eat like each other. Yeah, they do eat each other, and they can be aggressive. My first dissertation chapter was on looking at what they eat on artificial diets, and sort of really characterizing in a very in a lab environment what they eat. And one of the interesting things that came out of that was that the short wing crickets eat more protein than the long wing crickets. So they may have different preferences. Whether or not that translates out to Sedgwick, I don't know. Let's see.
We had an undergrad for a while who was feeding crickets little mushed up pieces of lettuce and mushrooms and various other strange things, and then dissecting the gut and looking under a microscope to see if we could find some signatures of the different foods, so that we could then do that on the wild crickets to find out what are you eating. But we haven't made any progress on that yet.
Oh, and in terms of how long they live, their generation time is a few months. It takes, like, when they're from a little hatchling to adult is like 2 to 3 months, depending on the temperature. But once they are an adult, in the lab, they can also live for another few months, but we think in the field, they only live for a few weeks. There are so many predators out here, not just flies, that like crickets. And so they're pretty likely to run into a predator pretty shortly. In terms of some other basic cricket questions, there was a question about temperature and being important for cricket song, and absolutely. The females like crickets that sing faster, and the warmer the crickets are, the faster they can sing. And so in terms of the males, their territory is really, really important for that reason. And the males hang out mostly in their little burrows. And so so in our temperature data, we found that those burrows were really, really hot. And so they were probably, those hot burrows are probably good prime real estate for males. Okay, let's see. And so yeah, so they make more noise if the temperature is hotter, because they sing a lot when they're loud. What other questions do we have? There are several questions about flies. And so some questions that came up is, why do we think the parasitoid flies spiked in 2018 specifically? And I wish I knew. I don't know exactly the conditions that are favorable for flies. In some of the prior research that has looked at the appearance of flies through the years, you can see a lot of annual variation. And I hadn't read anything about or nothing that I had read has sort of made any suggested hypotheses as to what conditions are favorable for the flies. The long wings, but yeah, may be able to escape better. And that was one of the year following the flies. I was kind of hoping that they were back because I had wanted to sort of see. I was kind of I had a pet hypothesis that maybe I would see a higher frequency of long wings the following year if they were better able to survive, but I didn't have any evidence to support that. What other questions? Do you want to take any?
I can't see them.
They're right here.
Oh, there's an interesting question about the general trajectory of flight being lost.
Oh, yeah.
So flight is a really expensive thing to do. It takes a lot of energy, not only to actually fly, but also to maintain the machinery that allows you to fly. So the flight muscles, the wings. And so it's really beneficial in certain circumstances, like when you need to be able to disperse a long way to find mates or new resources. But if those environmental selective pressures aren't present, if your patch that you're in is very, very good and stable, then the costs of flight eventually will lead to it being lost. And we see this repeatedly in insects that are on little islands, like offshore islands. Flight can be really dangerous in that case because you fly and then you might get swept out to sea. So in those circumstances, we see a loss of flight. But anywhere where the habitats are very stable and you don't need to disperse a long way to find mates and resources, then there's this repeated trajectory towards flight being lost. So we see a lot of flightless insects. You also see it in birds. So in New Zealand, where I come from, there's a huge predominance of flightless parrots because New Zealand evolved without any mammalian predators. We don't have any native mammals apart from 2 or 3 bat species. So the birds have repeatedly lost flight because the costs are great and the benefits are not big enough to cancel it out. So we think that having these 2 morphs is an intermediate step on a trajectory towards losing flight. Okay, the impact of temperature isn't that the one you already said?
That was not the one I was looking at. But how would the long wings do better in warmer temperatures?
Yeah, so what our work suggests is that cold temperatures at night are going to be limiting the ability to fly. And so if you're a long-winged cricket with these expensive flight muscles and wings and you can't actually fly, then it's cancelled out the benefits and the costs are there but not the benefits. So what we think is that thermal limitation on nighttime flight could be one of the factors that drives this evolutionary loss of flight in nocturnal insects. They can't go out and bask in the daytime like butterflies and dragonflies. And so this really strong limitation of their ability to fly at night might sort of drive the disappearance of flight as a strategy. And so we would expect, following from that long wings would be maintained longer and do better if temperatures are warmer because they can actually make use of their expensive flight apparatus.
Yeah. How does cricket behavior differ between summer and winter? I don't think we know. I think in the winter I think there's an overwintering stage, whereas juveniles, they just hang out and they don't do much.
Yeah,
but I'm not sure.
We don't know a lot about the overwintering of the crickets.
I've only been
very
out here
interesting to know. I expect they probably slow down, don't reproduce, hang out below ground a lot of the time, don't eat very much and wait for it to warm up again.
So you guys, I think to close it out tonight, there's been a couple of questions about the overall implications of cricket indicators of climate change. Do you want to close your talk with any observations about whether crickets are telling us something about climate change?
So we haven't really got there with the cricket system yet, although that's a direction that I'd like to go, because there are species of, there are 11 species of field crickets in California that sort of have ranges that span from Baja California all the way north. So I'd like to move out into a bit more of a broader geographic gradient to answer some of those questions about you know, on larger spatial scales and time scales, how are they going to respond to climate change? We might expect at the simplest level that as temperatures get warmer, it's going to be better for longwings, and that these crickets are going to be fine. So we don't have any indications from our work that they're going to be a species that is sensitive. So that's where I sort of switched, that this system is not as much about climate change as the other systems that I've worked on. But the big picture conclusions from the other insects that I've worked on are that, you know, we need to think about the energetic impacts of winters getting warmer. You know, as temperatures are increasing, overwintering insects are using up their energy reserves faster because their body temperatures are warmer. And this is leading to many insects actually dying or declining or not being able to reproduce because they run out of fuel, just like running the car with the pedal to the metal. So that's one of the underappreciated impacts. We tend to think about heat waves in summer, and that warming in winter is just going to be good because, like the mountain pine beetle, it's going to let insects expand north, which is, you know, maybe not good for us if we're talking about a forest pest, but generally good for insects. But I think the flip side of that is that warmer is not always better for an insect. Sometimes it's better to be cool because it sort of puts things on ice and keeps things running slowly. But the general picture that we're seeing is that really widely distributed weedy generalist species that already live everywhere are going to do just fine. But the majority of species who are, have their range confined to more specialized sort of climates or food resources, those are the ones that are going to decline. And that's why we're seeing this overall loss of insect biodiversity at the same time as we're seeing this sort of counterintuitive increase in like a lot of the things that we hear a lot about, which are the pest species. So we need to worry about those little sort of niche and little bit boutique species that are like our beetles living up in the Sierras that you might not see or hear so much about, but are so important to the functioning of their ecosystems.
I would just like to close tonight by thanking you for paying attention to the small things that, especially now with what's going on in the world, there's just not enough attention being played to the finer details of science, and insects certainly play into that. So I know you're heading back to New Zealand for the next couple months, and you're not allowed to stay there forever. We expect you back by at least next summer to resume your field work. Also, I've heard a rumor tonight that you're either going out to work in the field at Sedgwick, or your hotel in Solvang has a hot tub that closes. So,
The last, no more field work tonight.
You have 75 people still on this call that I think would love to be field assistants in the future.
Well, send me a message. We've had a high school teacher, Nikki Chambers, out in the field the last two years as a volunteer, so anything is possible.
So I really appreciate all the great questions tonight. I miss I miss those of you whose names I recognize more than you know, and you're always thinking and asking questions, and I appreciate your inquisitiveness and your sort of love of science. And thank you for stepping up and proving that you're still out there thinking. So we miss you guys, and thank you for Zooming in tonight. We'll come up with something. I feel like the next thing should probably be plants and not animals. In September, I feel like we've been a little animal heavy. And there's quite a few botanists that are Zooming in tonight. So I haven't forgotten you guys. And thank you to all the Berkeley folks that have stepped up. You guys have really fought hard to be able to continue with your research during COVID. It's not been easy in the UC system. You've had to jump through a lot of hoops, and we appreciate that from the Sedgwick level and from the university level. So with that, I will get this recording up on the Sedgwick website, probably not as fast as John and Liz will with the Natural History Society, but thank you again for Zooming in tonight, and we will talk to you all soon.
Yeah, thank you everyone. Thank you, Kate.
Good night, everybody. Thanks again.
Good night.